Well, thanks so much, Leon, for that introduction. And thanks so much for having me. Do let me know if my accent is impenetrable or if I'm speaking too fast or anything like that. So I think everyone in this room will immediately recognize these sculptures by Mathieu Lahanner as being the age structure of different populations. And I think also everyone in this room will recognize that some of the weirder distributions, like this is Qatar, with the very narrow bases, are the result of perturbations associated with declining birth rates and also patterns of migration. But of course, as the bubbles of individuals move up the age structure into the later years in all populations around the world, a really interesting question is, what shapes patterns of aging? Again, it will come as no shock to anyone in this room that this is what death looks like. Across age, there's very high probability of mortality among the young and high among the old, and you get what's called the classic bathtub shape in between. And as we march forward through time from 1922 to 52 to 82 to 2020, this bathtub has fallen lower and lower. So the probability of dying has fallen towards zero. And concomitantly, and as we'd expect, the life expectancy has increased as time has passed through. So the average number of years that individuals have lived. It no longer looks as linear as it did when Jim Open and Jim Bopel wrote a paper called The Broken Limits to Life Expectancy, which predicted a continuing linear increase forever. But it's still pretty striking how much life expectancy has increased. And I think what Jim Bopel, who died about a week ago, would say is that in the hour that we spend together, you will gain 15 minutes of life. Clearly, it matters. Understanding these processes matters. It matters as we move into the future, both these patterns of increasing life expectancy, but also the heterogeneities that underpin them. This green color is Japan, but what I had shown you, and without mentioning it, was the women, and women tend to live longer than males, and you can see that very strikingly in the patterns of life expectancy over age. And so we would like to understand this, and we need to understand this to predict what our populations will look like in the future, but obviously simply writing down the kind of statistical kicking things forward is not going to be enough. We need to dig into what underpins this, and this is in some sense the province of biodemography. So using biology to start trying to unpick aging, drawing upon the vast diversity of species that live on this planet, alongside the vast diversity of what we understand about us, to understand what our future will look like. There were lots of understands there. Understanding is good. There are three big questions that you might organize these teams around. The first is sort of, why do we age? The second is, how do we age? And the third is, when do we age? And many biodemographers and demographers for many years have pondered all of these three questions. And what I'm going to try and do is touch on all of them, but bring to it an immunological slant, because I'm very interested in how the immune system works. And I think it brings a slightly different perspective on every single one of these questions and represents some of the really exciting frontiers as we move forwards. So why do we age? You might be thinking, why are we even asking this question? If you are an evolutionary biologist, it is a really interesting question, right? As a very naive evolutionary biologist, you would expect that in order to maximize the representation of your genes in future generations, you would want to survive as much as possible and reproduce as much as possible. The fathers of, and unfortunately they were all fathers, of evolutionary biology, the ones that we know of at least, laid out the basic logic underlying this, and it's very straightforward. If you imagine a single cohort where the probability of dying is 50%, you start off with 2,000 individuals, by age 2 you might have 1,000 individuals, by age 3, 500, and so forth. So now this is our probability of dying. It's the same at every age. Let us imagine that there is an increase, a mutation that drives an increase in mortality around age 2. This is going to have a stonking effect on the population, right? Because you're going to reduce the number of 3-year-olds, and they're the 4-year-olds and 5-year-olds and 6-year-olds. And so you're just going to have this big reduction that trails forward through time. This matters. This will reduce your representation of genes in future generations. Conversely, if a mutation pops up that increases mortality at much later ages, and this population has a 50% mortality, so late ages are not very late, right, age 7, still, there's nobody there. So there's no discernible effect. So what we expect to happen is individuals that have a mutation that results in an increase in mortality at these late ages will have no detectable effect on fitness to a first approximation. Such mutations that increase late life mortality will increase in frequency in the populations. And this might be either through this process called mutation accumulation, which is just what happens. It's a result of drift, it's chance effects, but also potentially by antagonistic pleiotropy, which is just a fancy way of saying that some mutations that increase mortality at late ages might have benefits at earlier ages. They might increase survival, increase fertility at early ages. So there is incredibly robust evolutionary theory that tells us why we age. There are also funny things about this theory, right? It is funny to assume, I'm sure many of you in the room are thinking, that a mutation can act at a very specific age to have a very specific effect on mortality. In actual fact, that's not how living systems work. They're much more complicated machines, interacting machines with tons of feedback. And I think there's lots of really interesting space there for novel theory around what we expect the profile of late life mortality to be. And in particular, the immune system is a striking example of a system that's driven by feedbacks. Some recent evolutionary biologists have written some papers suggesting that it is actually the carry forwards of these systems that are set up to operate well early in life that determines what the shape of late life mortality looks like. So I'm not going to say much more on the why we age. I think the basic premise is there, but there's a lot of nuance that awaits unpicking and is very exciting. But we move from the why we age to thinking about how we age. So this is obviously the province of medicine and the province of epidemiology. There's a huge body of work there that I won't be delving into. Luminaries of this department like Noreen Goldman have done some beautiful work on looking at biomarkers that tell us about predictors of survival at late ages. And one of the interesting features of that work is that you find that in actual fact, asking people how well they do, how well they feel is often a better predictor of mortality than measuring a bunch of stuff in their blood, which is quite fascinating. But it also tells us that we need theory, we need to not just measure a bunch of stuff, but we need to actually bring mechanism and process to thinking about this problem. The biomarkers that we do find associated with immunity tend to be associated with inflammation. And there's this term, sort of slightly odd term, inflammaging, which refers to the fact that one of the phenomena that occurs at late ages is that your immune system is kind of constantly on a low level of alert. So there's sort of fluids seeping into your tissues and your macrophages are patrolling, and it's just not great for your health. But, and that's a very generic process, and that's pretty repeatable. So C-reactive protein, for example, you see in late age populations everywhere. But what if we took a more focused approach? What if we thought about parts of the immune system and how they develop over the life course in a little more detail? So for example, if we think about influenza, obviously a big killer of individuals at later ages. And also we used to think of it as the respiratory pathogen. We don't do that anymore. But it's this virus which has these two proteins on its surface, hemagglutinin and neuraminidase. And these are two things which our immune system learns to recognize. And by learning to recognize these features, it can repress the virus and protect us from disease or even infection in some cases. Now, there was this glorious cohort study that was done in Guangzhou province in China, where they went out and looked at a bunch of individuals across different ages. That's shown on the y-axis. And they looked at their antibody titers. So how many what was the density of antibodies in the blood of these individuals against different strains of influenza. And so the one on the left is the Hong Kong 68 strain, followed by Victoria 75, then Bangkok 79. These are all influenza strains that burst out of these particular, or were first detected in these locations and spread all around the world. And the really fascinating thing about this pattern is you can see that if you were, so these are the very young individuals, these are the oldest strains. The strains are going from the oldest to the most recent, ending in the 2008 strain. If you are too young, then you don't mount a titer to that particular influenza strain. This is not shocking, right? Many of us in this room would not raise a titer to Bangkok 68 because we weren't alive. There's no reason our immune system would recognize it. There's also this really fascinating pattern if you unpick what's going on under here, where it seems like your immune system becomes completely obsessed with the first influenza strain that it sees. So whatever titer you show it, it's still mounting a huge titer to the first thing you saw. And this shifts to, it's probably the thing you saw when you were around 7. So there's really interesting implications for what this means for what your late life protection will be, given that your immune system has sort of locked onto this early life pattern. But this is, of course, not mortality. This is just sort of, immune measurement. So what does this mean in terms of how do these antibody titers map onto how we die? So we also had associated with this data a biobank cohort study, so a nested case control study of 100 sex and age matched deceased individuals and living controls with baseline antibody titers to the different influenza strains. And we could use this to try and look at patterns of survival associated with having particular antibody titers. And this was kind of instantly surprising. So I'm going to plot it out this way with, this is the different years of circulation of the virus strain, and this is the odds increase in mortality. So if you're above the line, then you're more likely to die with an increase in antibody titer, and if you're below, you're less likely to die. And the pattern was like this. And I want to say this was a preliminary study. It was not powered for significance, but the effects are still striking, because you're seeing this 20% almost decrease in mortality with an increase in antibody titer. And this is shocking because it just can't be that there's that much flu, right? 20% is a huge number, and influenza is not killing that fraction of the population. So there must be something else going on. And what's even more intriguing, if you dig into this a little bit further, is that if you break out males from females, you find sort of opposite patterns. It seems like antibody titers are protective in males, but hurt females. Again, nothing is significant. We're writing grants to try and repeat the study at larger scales now, but it tells you also that what you think about biomarkers and what biomarkers are doing might be extraordinarily context specific. And this hints at something that's really important when you're thinking about the immune system, which is that it is a very dangerous line of defense. The thing that kills you quite often is not the pathogen, it's your immune system flailing around trying to defend you, and that's why you die. A striking example of this, of course, was the SARS-CoV-2 pandemic, and this is another example where patterns of infectious disease and immunity are intersecting with patterns of aging in an extremely striking way. So this is data from Public Health France, showing the number of hospitalizations, ICU patients, and deaths across age with, I think it was females on the top and males on the bottom. And that does make sense because females would be lower. And clearly the most striking thing that bubbled out of the pandemic was this pattern of greater vulnerability at later ages. And so we thought, well, it's interesting, but also, one thing we can do is put this in the context of what we see for other infectious diseases. Was there something weird about SARS-CoV-2, or did it look a lot like other infections when it came to translating infection into death as a function of age? And so what we did was we plotted out the relative risk of hospitalization. We also did ICUs and deaths, but I'm just showing you hospitalization as a function of age, standardizing by whatever the level was at age 60. So I'm showing you the influenza mortality and all-cause mortality, and they just look like the classic bathtub, right? And then what I can do is I can take the patterns that we have for the other coronaviruses, and we didn't just have SARS-CoV-2, we also had Middle Eastern respiratory syndrome, which is a pathogen, another coronavirus pathogen that has affected people mostly in Southeast Saudi Arabia, but has also spread to some other parts of the world. And probably the shape of the graph has somewhat tipped you off, but the pattern that we get is something like this. So the patterns really map onto each other quite closely at late ages, and they look a lot like the broad pattern of aging. Whatever getting infected is doing to you at late ages looks a lot like whatever infecting is happening to you for influenza or other diseases. And again, you get more vulnerability in the younger ages, but you also get this very striking, the whole line starts much, much lower. So it looks a lot as if, in some sense, that pattern of senescence, that breakdown of immune functioning and protection, is happening much lower for these emergent pathogens. And the big difference, and in fact, the only real difference between SARS-CoV-2 and MERS and the other pathogens that are circulating sort of in general, and that, for example, influenza shown here, is that we've never seen them before. So we have no immunity to them. It seems like what this adaptive immunity that the other infections are benefiting from is protecting them and resulting in this bathtub shape. So if you experience influenza during your middle years, if you're just about anyone on this planet, you almost certainly would have seen it earlier in life, and your immune system will have learnt to protect you against it. So kind of an interesting effect there of how how we have to think about adaptive immunity also as something which learns, so which also doesn't map onto the simple idea of a mutation resulting in an increase in mortality in late ages. So, gone through an example of why, talked a little bit about how the sort of clashing between different biomarkers and different infections might translate into different patterns of mortality. There's also the province of when. When do we start experiencing this uptick in mortality? And this is something, of course, that pattern with coronaviruses had already hinted at, but we can dig into it a little deeper. And the first thing, of course, people did within the classic domain of biodemography was to step back and ask what the patterns of mortality looked like when we lay them out across the tree of life. And I'm not sure you can see this very well, but this is a huge number of different species found across the planet, a big review in Nature that looked at their patterns of mortality over age and their patterns of fertility over age. So I can't read these either, but it's everything from monkeys to whales to tortoises. And you tend to get this pretty consistent, many species show an increase in mortality with age, but for some of them, it kind of flatlines. It depends on whether you're something which has indeterminate growth or not. Descends on a whole series of different patterns. So alongside these efforts that have worked to dig into what the pattern of aging looks like more broadly across the tree of life, there has been some lovely work also that puts the pattern of our aging, so the pattern of when we die, into the context of our nearest relatives, which are, of course, a whole bunch of different primates. And there, again, you see a sort of pretty consistent difference between the sexes as we have in our own species, and you see some subtleties and differences in when the mortality happens. So this is interesting and can help us to start thinking about the broad evolutionary drivers that might push around when mortality starts to go up in different species and different times. But of course, what I'm interested in, what I really like thinking about is not just this in general, but how we might think about this in the context of what the immune system does. And this is really the crux, I think, the nitty-gritty of some of the funkiness here is that the immune system is incredibly prone to long-term programming. So things that happen early in life have very long shadows in the context of thinking about immune function. There's an extremely easy example of this, right? So if you just think about any directly transmitted infectious disease that is immunizing, like measles. So this is an illustration by Sophie Blackwell showing a child brushing measles away, because there are huge public health efforts underway to get rid of measles because it is such a huge public health burden. But you can model measles very easily by using a very simple schematic where individuals are born, susceptible, they become infected, on being infected and on encountering an individual with measles, and then they recover. And you can see that the very fact that the arrows only go one way and there's no return tells you that what happens early in life really matters. If you have been infected early in life, then you will be protected for the rest of your life, as far as we know. And what's even better is that we have available a safe, inexpensive, and completely immunizing vaccine. The vaccine is so safe and so inexpensive, and the disease can be so deadly that the measles vaccine has often been referred to as a best buy in public health. And what that does is it moves you, if you vaccinate individuals around birth, it moves you straight out of the susceptible box and into the recovered box. And then you're protected, as far as we know, for life, which is great, right? So this is, but it also speaks very strongly to the repercussions of early life exposures for long-term outcomes in the light of immunity. Some lovely work by Amy Winter, who was a graduate student at OPR a number of years ago and is now a professor, an assistant professor at UGA, looked at circulation of measles and the impact of measles vaccination in Madagascar as part of her PhD. And Madagascar is a setting where the vaccination was introduced into the 80s, but kind of really took hold around the 2000s. You can see this is vaccination coverage going up. And then measles incidence kind of bopped around for a couple of years after vaccine introduction and then fell precipitously. And there was a very long period with very low incidence of measles infection in Madagascar. And what Amy did was she came to this data with a sort of fairly deep concern, which is that just because you see no circulating infection or very little circulating infection, that doesn't necessarily mean you're in a very good place, right? For measles, it could either mean that you're doing great and you're vaccinated a bunch of people and so everybody's protected, or it could mean that you're not vaccinating as well, but there isn't that much circulation. So the number of susceptible individuals is growing and growing and growing, and you're at risk of a really ginormous outbreak. And Amy wrote a paper during her PhD, which laid out that leveraged some samples that had been collected as part of the fever rash surveillance in Madagascar, tested them for an antibody titer that indicates whether individuals are protected from measles or not, and looked at the age profile of protection. And what she found was that there was a massive pool of susceptible individuals, suggesting that the vaccination program was not as effective as one would hope. And what she did was built a series of models to predict what the risk of a measles outbreak was. And because we don't know all the details, this model construction entailed a vast amount of sensitivity analysis. But under every variant of sensitivity analysis that she did, she found that there was a tremendous risk of a very big outbreak in Madagascar. And she published this paper. It came out in early 2018. And very sadly, in late 2018, the outbreak took fire in Madagascar. There was a huge surge of cases and many children died. So an example of where the early life consequences of measles infection can be really, or the longer term consequences, both at the individual but also the population scale of measles immunity are really quite drastic. There is another question. So this is sort of the very generic 0-1 in terms of protection. There is another really interesting question, which is, I've framed it in terms of you're susceptible or you're infected. In actual fact, the readout underlying this is the level of antibody titer, so how much titer you have in your blood. And this is a continuum, of course. And another graduate student who is currently in the Department of Ecology and Evolutionary Biology realized that one could use this sequence of events, right? So the fact that there was a fall in measles incidence, there was 10 years with very little, and then there was a huge outbreak, to look at individuals who'd grown up in different backgrounds of measles circulation, to understand whether actually just living amongst a population where measles was fairly frequent was different from living in a population where measles was very rare. And this is the world we're moving into, right? We're moving into a world where many infectious diseases are very, very rare. So any benefits we were getting from boosting, right, just being around individuals who had the infection, which would, might lead an uptick in our protection without getting us infected. So it'd be subclinical, but it would keep our levels of immunity high. We could be losing those, and we just have never seen this world before, so we don't know much about what it will look like. So what Arthur did was he pulled together cohorts of serosamples from kids who'd grown up in this phase where there was a lot of measles incidence and kids who'd grown up in a phase where there really wasn't, and plotted out their geometric mean measles titer. And what he found was that for children who lived in a high measles circulation era, their titers were raised probably by vaccination, perhaps by infection, and then stayed high. But if you were living in an era of low measles circulation, at some stage your titers started dropping off. Now, there's no cause for panic, I should say. These numbers, they're around 3,000. The protective threshold for protection from measles infection is thought to be about 200. So there's a really long way for this to drop before we start worrying about it. But it's more the generic pattern is interesting. This is, relatively a recent thing that we have started to document. There's one other paper that shows this. It doesn't seem to be a big deal for measles. But of course, we have to think about all the other infections that are out there, that we're starting to understand that the immunity that any individual has is a function of the immunity in the population around them, or at least could be. And that means as we move into a world where vaccination is wonderfully more ubiquitous, we also move into a world where immunity could be fading in ways that we don't understand. And we probably should try to understand. So this is sort of very much a pathogen-focused side, right? So pathogens circulating, the immunity we develop to them early in life, this long shadow over late life, this population consequence of circulation of infections. But increasingly, it is recognized that it's not just, as we have speciated, us and all our vertebrate colleagues is the wrong word, co-planetary inhabitants, leading to this beautiful diversity on the planet. It's relatively recently been recognized that alongside us, there's been a co-speciation of microbes, and not just microbes that are bad for us, right? They have co-diversified, they have transmitted between generations between us. Some of them are really just present or absent, and some of them are actually really important for our health. Okay, and so we have evolved in a context where we have commensals, where we have mutualisms with all these microbes, and of course these microbes too are disappearing. And if we are to understand the long shadow of immunity and our health in late life, this is something else that we need to grapple with. Just to give you an example of how striking this is, there was an experiment done with mice where they took a mouse and they put it in really cold conditions so that the mouse might start thinking about hibernating. And then they took the microbes out of the belly of that mouse and put them in a different mouse that was not kept in a cold temperature, and it started preparing for hibernation. So it's reading its cues off the microbes that it has, and presumably this is true of us too. Presumably a lot of our physiology and metabolism is geared to register signals from the microbes that are around us. And this actually, if you're an evolutionary biologist, this makes so much sense, right? We have an immune system that is exquisitely evolved to detect microbes. They probably contain a lot of information about the environment that you're actually easily evolved to pick up. So you would expect these sorts of dependencies to evolve. This poses actually a really interesting problem for the immune system, of course, as well, because some of these microbes are wicked and some of them are good, and they look a lot like each other. And so the immune system has to somehow be able to discriminate between the goodies and the baddies when the goodies look a lot like the baddies. And one of the ways this seems to happen speaks also to the ways in which early life immunity results in very interesting long life patterns. So there's a series of experiments done in germ-free mice where they take a mouse. If you take a mouse and you keep it germ-free, which is a very hard thing to do, but technology was developed a few years ago, then you find that this mouse is susceptible to asthma. If you take that same mouse and you show it a microbe or two, like a day or two after 2 weeks, it's still susceptible to asthma. But if you show it those microbes before the 2 weeks are up, then the lung develops in a healthy fashion. And there's one more, I forget exactly. But if you show it microbes sort of within this window, microbes or microbe-associated components, you end up with a healthy lung. So it's like there's this window, this very narrow window of opportunity where your immune system learns to be friends with some microbes. And if it doesn't see microbes in that window, then you're going to end up with asthma, which is your immune system attacking you, right? It's overreacting to you or to your commensals. It's overreacting to things that it should not be reacting to. So this is also kind of quite fascinating if we think about the context of where we sit in the planet and how microbes are disappearing. Everything from cesarean sections to delivery of antibiotics to children early in life will be perturbing this sort of relationship. And as I said, this could have a very long-term implications. This could be resulting in, obesity later in life. This could be resulting in different chronic diseases. It's clearly something that we need to drill in and better understand. Because I'm an evolutionary biologist and I love trying to think about, why things look the way they do. The first thing that titillated me about this pattern is just how crisp the cutoff is. 2 weeks precisely. And there's a series of different adaptations that lead to that. So I was led to probe the question of how long should this window be? It's accept that you need that window, then what might shape how long it should be? And I think it's, when you need to learn tolerance, but there are balances in how you think about, how long that window would be. It would have to be long enough so as you could see some of the good microbes, but it can't be so long that pathogens can sneak in there. So there's going to be a balance that is entirely determined by, or at least not entirely determined, but somewhat determined by the ecology of the microbes that surround you. But it's also, of course, going to be determined by the particulars of your different life history. And so, it might depend on your longevity as well as your exposure to microbes of various sorts. I wrote down a pretty simple theoretical model to interrogate this question, and you can make a series of predictions about when you would expect long versus short windows of tolerance and how they might be pushed around. Just to give you one example of this, You can look at the question of magnitude of seasonality, and this is something where microbes, for example, in monkey populations, particularly monkey populations that live on trash heaps, have been shown to be tremendously seasonal. Monkey populations, of course, that live on trash heaps are a very interesting model for the development of obesity, because they're eating the same crap that we are, and so they get fat. And so magnitude of seasonality, you can go from like almost no seasonality, sort of flattish, to extreme seasonality. And you can ask how wide this window of tolerance should be. And it probably won't shock you to learn that it needs to get wider as seasonality gets stronger, because there's a chance, if you're born at a time when there's very few microbes around, you need to extend that window to be sure that you pick up some of those microbes so that you learn to tolerate them. This is if there's no pathogens around. If there are pathogens around, you crunch down the window. So this is one of the things I like about this is it gives us a way, instead of just sort of, describing, here's a bunch of biomarkers, this is what survival might look like, we can start thinking about the processes that might underpin these different pieces and that might translate into patterns of mortality and so forth. Of course, this was I built a very simple-minded model because I'm a very simple-minded person. But my wonderful postdoc, Burcu Tepikula, who is an engineer by training, is building a much more intricate, much more detailed systems model of how immunity works at these early ages. And she has this beautiful understanding of all the different pieces, and it's just fascinating. So it seems, for example, that one of the things that happens is that you get antibodies from your mother that coat bacteria. And if bacteria are coated, that means that they're kept just far enough away from your gut wall so that your immune system doesn't attack them. And they get put onto a different sort of train tracks that keeps them in the coating domain. But if your mother hadn't seen them before, they go onto a different railway track that puts them into the recognize and neutralize. So there's these multi-generational potential effects of what you have learned to tolerate that comes from what your mother had learned to tolerate that we understand in increasingly exquisite mechanistic detail. So we should be able to make models of what we expect from perturbations and then perturb our models to see what other perturbations might do. This is at the very kind of both theoretical and mechanistic end. At the other end of the scale, Gugu Moyo, who is here, has been doing some lovely work thinking more deeply about the long-term repercussions of these impacts. So we know quite a lot about, what the demography of the planet in general. This is starting to tell us something about what we might expect, since we know things like how much antibiotics is distributed, how frequent are cesarean sections. We can start looking to lay out the shadow of these impacts in terms of mid or late life diseases on populations. And Gugu is particularly interested in what's happening in sub-Saharan Africa, where we do think that things like, the obesity epidemic, all these things are likely to strike alongside these impacts of disappearing microbes. And so it makes sense to start thinking about what we expect for the demography and how different perturbations, this is completely made up of things like antibiotic use, might be affecting the burden of different diseases. So I hope I've given you a few sort of examples of how both immunity has really striking impacts on patterns of health. I mean, that's not shocking, right? But interesting intersections with how this plays out across age, but also interesting linkages between what happens at different ages. And I think it's a really exciting time to start thinking about immunoepidemiology, both because of this, but because we're beginning to have the tools where we start thinking about the underlying mechanisms. And this matters, of course, from these lofty and fascinating goals of trying to map out the future of aging. The other side of the coin is, of course, this matters enormously if we're thinking about pathogens and pathogen dynamics. So Brian Grenfell and I have been talking for a number of years about how enormously valuable it would be to have something that we refer to as a global immunological observatory. And this is the fact that, I could go out into this room right now and take blood from all of you, which would be extremely creepy. But I could then figure out, I would know, we now have the technology such that I would know if you had been exposed to any of those flu strains I described, to if any of you had ever been anywhere near Ebola, if you'd seen what particular enteroviruses you'd seen. Only 200 and some viruses have ever been known to infect humans, and you can put all of them on a tiny chip. And so for the drop of your blood, I could know which you had seen, or begin to walk towards that at least. The technology is still under development. And this is both incredibly valuable in terms of understanding our past, right? For example, we would learn that most of us in the room had not seen Bangkok 68, which would not shock us because we're not that old. Some of us might have seen it. But we would also, there might be things that we'd seen that were surprising. So there are new enteroviruses emerging all the time. And you might, we'd be like, oh, that's kind of odd. Like maybe we should dig into that. So it tells us something about the past. It's this incredible window onto the past, but it also tells us about the future, because of course, we are the substrate on which these pathogens spread. And if we are immune or protected from different pathogens, then they cannot spread. Everybody in this room, I have no doubt, has very high antibodies to measles. Strikingly, actually, when I went for my green card, it turned out I did not have very high antibodies to titers to measles. But to a first approximation, it's extremely unlikely that measles could spread in this community. But we'd like to know, and there are settings where that is not the case, and the settings where it could be quite straightforward to get to grips with this. There are huge convenience samples that are lying around, so there's everything from blood banks to collections for malaria testing, for HIV testing, that you could repurpose to get a sense of what this landscape looked like. I think, there's a clear cause for it in terms of understanding the dynamics of pathogens. There's also real vast interest in getting a better sense of the age trajectory of our immune systems, both in this context I referred to as the disappearing microbes issue, but also, everything to do with chronic diseases expanding. The ways in which we die are increasingly associated with our immune systems attacking us. So being able to build or understand how that builds itself over our lifetimes is going to take longitudinal data on how that immunity spreads out. We also have, and this is, and there's been some spectacular work. I just want to mention many fabulous people who've worked on this. Rachel Baker, who was a postdoc in our department until recently, and she's now a professor at Brown, Daniel Larimore at Colorado, and Daniel Park and Arthur Menzies, who are graduate students in EEB, and James Hay, who is at Oxford now, all have been putting together the kind of statistical and mechanistic modeling machinery to try and make sense of the sort of data that you might get out of this sort of data bank. And there also, we have some wonderful colleagues at NIH who are pulling together a set of samples of exactly this kind, where they're measuring just about everything in a bunch of different people from locations all around the world. So this is an effort called Premise. And all of this should give us, the best, the absolutely most exquisitely sensitive and detailed sensor we have to infectious diseases is our own immune system. So this is a way of harnessing it to figure out what the world looks like. And then we would, if we knew what normal was in terms of immunity around the world, we should be able to detect anomalies, which might also alert us to things like the emergence of novel pathogens, which turns out it's an issue. There's also some really interesting, I think, opportunities recently. So all of you may remember that we had 2 or 3 years where we had absolutely no influenza. This is again some work by Rachel Baker, where she was looking at, this is influenza cases in all the states of the United States, showing you surges. I think this is relative to background. And once the national emergency was declared, you can see that influenza dropped off the map. Not shocking, nobody was talking to each other anymore. Couldn't give each other influenza either. But this means we have this super fascinating situation where there have been, on the one hand, the potential for the absence of cases means you could get a buildup of susceptible individuals, because it is being infected that reduces susceptibility in the population. So it makes sense to sort of, think about this very deeply in terms of thinking about infectious disease dynamics, but it also gives us a window onto what happens to our systems as we are not exposed to infectious diseases at regular intervals. There's a parallel effort which Daniel Park is driving to, which is looking at D68, which is one of the enteroviruses that emerged in the States relatively recently, has biannual cycles, has an outbreak every 2 years. An outbreak every 2 years is a very strong indicator that this infection is immunizing. We predicted that after the pandemic, you should have a year or so of honeymoon where there wasn't much going on, there wasn't much transmission, and then a big surge. And you could build models to predict this. And the models did really well at predicting infection. This is very tragically a disease that also, like polio, occasionally paralyzes children. The really surprising thing here is that although we've been very good at predicting infections, we have not been very good at predicting disease. There has been far less disease than we expected. which implies that there's something subtle going on, maybe about, what order you get infections in, how old you are when you get infections. And it is only perturbations like the phase of non-pharmaceutical interventions during the pandemic that will give us a window onto that sort of process. So Brian and I, with Chelly Sadroy, a very talented graduate student of Brian's who's now at Berkeley, have been calling alongside this to a study of immunoepidemiology. We need to understand both sides of the system. It is our immune system that chases the viruses around the landscape. They evolve to escape our immune system, and they come back and hurt us more. And it's a great glorious cycle, great not glorious cycle, it's a great tragic cycle. There's just a whole suite of data that we could bring to bear on this problem that would put us in a much better place in meeting both the challenge of emergent pathogens, but also how we grapple with endemic pathogens in our populations. And of course, so this is something, I think the infectious disease side is completely and absolutely obvious, but what I hope I've also opened the window onto a little bit is the side in which this also sheds light into patterns of how we age over time and the ways in which the patterns of health that we experience will be shaped by the patterns of exposures to different pathogens that we experience and different microbes as well, and microbes in general, and how we should really start thinking about what it means to be growing up in this world, to be aging in this world that is not like any world we've seen before, in part because we don't have the microbes that every other generation has had. So that's what I've got. There's some of the papers, and thank you very much.